Hello, everyone. Thank you all for joining us for our November Research Spotlight webinar. I'm Dr. Sarah Hernandez, the Director of Research Programs at the Hereditary Disease Foundation. I want to start by thanking our sponsor for this webinar, uniQure, and also thank everyone who supports HDF, and to all of you for tuning in. Thanks for being here. I'm excited about today's webinar because I get to introduce you all to someone that I used to work with in the lab. She taught me everything I know about working with stem cells. I also had the pleasure of spending a few days with her just a few weeks ago at HDF workshop in New York City, focused on young investigators, where we had 24 HDF-funded young investigators gather to discuss their research, share ideas, and generate inspiration. We are joined today by Dr. Charlene Smith, a project scientist in the lab of Dr. Leslie Thompson at the University of California, Irvine. Dr. Smith carried out her PhD studies at Cardiff University, helping to develop a protocol that turns stem cells into a specific type of brain cell called medium spiny neurons. And these are the cells that are most vulnerable on HD. And actually, it's this protocol that Charlie helped to develop that is currently being used by many labs around the world that study Huntington's. Dr. Smith then applied her protocol to a postdoctoral work in Dr. Thompson's lab to model HD in a dish so that she could study biological changes that are caused by Huntington's. She identified differences in these HD neurons that are generated from stem cells, specifically in the way that the cells develop and in structures inside the cellular powerhouse called the mitochondria. Dr. Smith also developed a strategy using CRISPR to lower levels of a protein called PIAS1. And she has found that lowering levels of PIAS1 in these HD neurons derived from stem cells has protective effects. She's now continuing this work in 3D mini brains. Dr. Smith has received an HDF postdoctoral fellowship. She is active in HDF young investigator meetings. She contributes to HD community groups, and she served as the chair of the Gordon Research Seminar for CAG triplet repeat disorders in 2019. We are excited to have you with us today, Charlie, so thank you so much for joining us. Before we start, I want to remind everyone that you can ask questions in the Q&A box throughout the talk, not the chat box, the Q&A box. We are going to have HD families as well as researchers on the call, so feel free to ask both technical and non-technical questions. The important part is just ask them when you think of them so that you don't forget. We should have about 25 minutes at the end to get to a Q&A, and we're going to try to address as many of those as we can. So again, thank you all so much for being here, and I will pass it over to Charlie.
Thank you, Sarah, for that really lovely introduction. And I'm very happy to be here today to talk about my research. It's been a fairly short career, and hopefully I'll be able to summarize all of that for you guys today. And so I'll go ahead and share screen. I'm going to talk to you about brains in a dish and using stem cells to advance Huntington's disease research. So before we get started, what are stem cells? Stem cells can form any cell type of the body. This is what we term pluripotent. They can also divide indefinitely, so they're an endless source of cells for us in research, which is a great advantage for us to have. Additionally, there are adult stem cells. These are what we call multipotent, and they are more limited in the cell types that they're able to make. And so the reason that stem cells are pluripotent is because they've originally, they are able to form all cell types of the body, because that's kind of like the cells that we have in the developing embryo. So just going through this little diagram on the right here, we can see that we have this ball of stem cells here. And then if we go on, they can take one of those cells, and then we can either divide it to make more stem cells, or we can start differentiating it or start patterning it to become a certain type of cell. So for example, this cell here on the left in red has been a little bit more specialized at this point, and then can go on to form, for example, some other cell types, such as the muscle or other cell types. This cell then on the right can go on to divide even further to form more stem cells, or go a little bit more specialized, say for a cell type that will then go on to say form a neuron or a skin cell. So how we make stem cells for HD research is one of two ways, really. We can either get embryos that have been discarded, usually from IVF, and use those to take cells from the inner cell mass, this little part of the cell, the cells in the embryo. and take those and culture those, and those kind of then become pluripotent stem cells. Or the other way is to take either a blood biopsy, blood sample, or a skin biopsy from under the armpit to be able to take skin cells that haven't been exposed to UV light. And then we can treat them with these 4 factors, c-Myc, OCT4, KLF4, and SOX2. And these factors can kind of like reprogram the cells to be more like pluripotent stem cells. And this actually gets them to a really good pluripotent stem cell source. And these are called induced pluripotent stem cells because they've been told to become stem cells again. So the HD-iPSC consortium was formed by the NIH and these investigators, one of which being my boss, Dr. Leslie Thompson. And this was formed back in the the 2000s. And then CHDI helped fund a little bit further research so that they were able to distribute these cells that were generated from patients, and be able to form more cell types and distribute these cells, to other labs. So they were able to work on them and see different things that we can find out from these stem cells. And if we can make them into neurons, the cell type, one of the cell types affected in Huntington's disease, whether that would shed any light on what goes wrong in the disease. So what over the years we've been able to make with stem cells, we're able to make a lot of different cell types. We can make either mixed neuron cultures. So these are like a mixture of neurons, so the brain cells that communicate with each other, or you can have like astrocytes and progenitor cells in there as well. Additionally, we can make cortical neurons, so the cell type that is also affected in Huntington's disease. We can make part of the blood-brain barrier called the brain microvascular endothelial cells. We can make organoids. So what we call, these are kind of like more 3D, what some people term mini brains, but they kind of replicate a lot of the structures that we see in the brain, but are not completely like the brain. We can make neural progenitor cells, medium spiny neurons, which are the cell type that are most affected in Huntington's disease, and what I spent a lot of my time in my PhD studying how to make. We can make microglia, which is kind of the immune system of the brain, as well as astrocytes, which are very much a supportive cell of the brain, but help do a lot of functions in regulation as well. So what we've learned about HD with stem cells is that neurodevelopment can be affected in Huntington's disease, as well as how the cells communicate with each other. So either the communication at the synapses, which is where 2 neurons come together, and there's communication between those 2. We've seen that there's dysfunction there. We also see there's dysfunction in calcium signaling, so kind of again how the cell can communicate with each other and regulate itself. We've seen that there's issues in the mitochondria, which is the powerhouse of the cell. So this is what produces energy for our cells to be able to use. We've also seen that there is aggregation and accumulation of Huntington protein in some cells, some neurons that have been differentiated from stem cells. Sarah Hernandez here and Ryan Lim, a former grad student in our lab, was able to show that there is issues with the brain microvascular endothelial cells, so part of this blood-brain barrier in HD. And also there is issues in the nucleus. So we have issues with transcriptional regulation. So the genes that are expressed in HD-iPSC neurons or iPSC-derived cells are different to unaffected individual cells. We've seen that there's issues in DNA damage repair. So there seems to be an accumulation of DNA damage in these cells. There's issues with nuclear cytoplasmic transport, so transport of proteins or things between the nucleus and the cytoplasm, which helps for the cell to function, as well as differences in these marks on DNA, which kind of help with the expression of genes as well. So I kind of want to take you back in time now to show you why I study Huntington's disease. So we're going to follow this kind of timeline on the left of my kind of career and journey through being an undergrad. when I learned about this lady, Dr. Nancy Wexler. I had a lecture at Cardiff University where Professor Anne Rosser told us about Huntington's disease, and then started talking about the gene hunters and how they were trying to, they went to travel to Venezuela and were able to try and figure out basically what was the gene that was causing Huntington's disease. And I'm not sure what it was about that lecture, but I was hooked. And so I kind of deviated my path a little bit and thought, okay, maybe I'll go and study at grad school and try and study Huntington's disease. And what we were trying to do at the time when I was kind of in undergrad, it seemed like a lot of the research, like we just learned about induced pluripotent stem cells, so the cells that you can kind of reprogram to be pluripotent. And we were then trying to make the cell types that are most affected in Huntington's disease the medium spiny neurons. And at the time, the landscape was very much, we can get about 10% in our dish of neurons. Like we can get 10% cells that look like medium spiny neurons. And so these were a lot of great papers at the time, really pushing the field and trying to push to get this high percentage of medium spiny neurons. But it seems that there was this limit. And so at the end of my undergrad, I was finishing and I was looking for a PhD in grad school. And I joined up with my two PIs, Professor Paul Kemp and Professor Nick Allen at Cardiff University, who together were part of the iPSC consortium for HD-iPSC consortium with CHDI. And so I tried to make medium spiny neurons. And the way I was trying to go about it was by using some data that was published from Dr. Miriam Hyman, who had shown that if you profile the cells that are in the striatum, so there are these two types of neurons in the striatum called DRD1 and DRD2 neurons, and they're just expressing these proteins differently, and they are part of the different pathways of the striatum. And she profiled these for the genes that were expressed in those cells, and we thought, okay, well, if we maybe kind of use a similar idea of the induced pluripotent stem cells, of expressing these genes in the cells that Miriam had found were in these DRD1 or DRD2 neurons, that we would be able to maybe make medium spiny neurons. And so this was quite successful. We were able to make medium spiny neurons, and we look at this by looking at these expression of these two proteins, one in red, DARP32, and one in green, BCL11B. And we look for co-localization of that to see if they are, in fact, a medium spiny neuron. And so we were able to do that, but it was kind of an inefficient method with how I was doing it, just with plasmids and not using like maybe viral transduction. And so at the same time in the lab, we were trying to make neurons that were electrophysiologically active. So what I mean by that is these cells are able to communicate with each other. And the way that neurons tend to communicate with each other is through electrical activity. And we were trying to figure out a way to make these more electrophysiologically active. So a lot of the grad students in the lab were helping, and as well as the postdocs, to try and figure out how's the best way to make these electrophysiologically active. And we were very much an electrophysiology lab. And we then published this paper showing that if we can push the cells out of the cell cycle, which is how the cells keep dividing, then we can make them post-mitotic neurons, we term that, and that actually pushes them to be electrophysiologically active, as well as treating them with a few other drugs to help them. And so this took a long time to do, and so I actually graduated from my PhD without having worked on HD, even though we were in a lab that was very much working towards HD and HD models. And so I kind of spoke to my PI at the time and was like, okay, I really want to work on Huntington's disease, so where should I go? So he was like, you should go work with Leslie Thompson. And so I got on a plane, flew to from Cardiff to Irvine, and then a few weeks later, my dogs joined me. So they, it was a few weeks without them, which was kind of sad, but we was able to get into the lab, get over the jet lag, and then I started working on Huntington's disease. So while what I did during my PhD worked well for making the medium spiny neurons with these transcription factors, like in trying to push them into medium spiny neurons, like I said, it wasn't very efficient. And so we thought we were working to try and push them to be more medium spiny neuron-like. And at the same time, collaborators that were at Cardiff University, Dr. Meng Li, found that if you add this one drug, then you could potentially push them towards a striatal neuron phenotype, so more like the medium spiny neurons that are affected. And so we thought about adding this into our current protocol and found that it does actually increase the number of this DARP32 BCL11B in red and green again, seeing that there's an increase in this percentage. And this is the kind of change we see. And I forget to mention, sorry, that this here on the left is how the lab looked when I joined back in 2015. So it was a little bit smaller than we are right now, but still a great bunch of people. And so bringing this protocol to UCI, it involved a couple of different steps. So we have the stem cells at the beginning, the iPSCs. We then push them towards a neural stem cell phenotype, and then neural progenitors. And then we kind of just pushing them even further and further to make them so that they're postmitotic medium spiny neurons. We have a lot of iPSC lines at UC Irvine that we've had access to as a result of the HD-iPSC consortium. And so we have a lot of unaffected individual cells, which are very important because we need to have something to compare to. But we also have a lot of adult onset lines as well as juvenile onset. And so we wanted to combine like the expertise and so we wanted to try to teach, like Sarah said, I taught everyone at UC Irvine the protocol, and this is me showing Jennifer Stocksdale, a grad student in our lab now, how to make the medium spiny neurons. And so we wanted to kind of, now we have this protocol that makes a decent amount of medium spiny neurons. We wanted to find out, again, what goes wrong in Huntington's disease. So we explored this transcriptional dysregulation and epigenetic changes in HD-iPSC neurons. And so this is a project that Dr. Sarah Hernandez worked on with me very closely as well. And we made the cells into, we used a lot of our HD patient lines as well as our control lines. And we made them into the medium spiny neurons, as you can see here with this DARP32 BCL11B expression in the red and green on the left. They also express another protein called FOXP1, which is another kind of striatal marker, a medium spiny neuron marker, and MAP2, which is a neuronal marker, as well as GAD65, another MSN, medium spiny neuron marker. And so with that protocol being established across all of those lines, we then put them through a battery of tests. So we did RNA-seq to have a look at the transcriptional changes, as well as ChIP-seq to kind of look at the similar, whether there are marks on the DNA that make us think that there is a transcription happening. We also looked at flow cytometry to assess certain cell types that we were finding, and finally we went on to perform single cell RNA-seq, so looking at the different changes of RNA expression, so gene expression, but on an individual cell level. So firstly, what we found was there was epigenetic changes and transcriptional dysregulation observed that highlighted the cell cycle. So just to go into this data a little bit, we found in RNA-seq, so we saw that there were 700 changes between our HD samples and our unaffected samples between the gene expression changes. When we looked at the ChIP-seq, so looking at the DNA marks, we found that there was over 1,000 of these changes. And there was actually a really good overlap, around about 327 of these DNA marks and gene expression changes were overlapping. And what we found with those is a lot of these genes basically represent changes in the cell cycle. And this was work in collaboration with Ernest Frankel's lab here. Their lab performed a lot of the ChIP-seq analysis. And so this was kind of rather concerning to me. I'd come all the way from Cardiff to Irvine and like with this protocol thinking, well, we make these neurons that shouldn't be going through the cell cycle, they shouldn't be dividing. What's going on here? And so we kind of looked into this a little bit further, and I took a step back and looked into the literature. And so there has been previous work that shows that there's maybe things that go wrong maybe with neurodevelopment, or there's changes in the ability of the cells to divide in a Huntington's disease. A paper from Richard Fall's lab showed that there was an increase in PCNA expression and in the subependymal layer of the brain. And this changed with the grade of HD brains at postmortem. Larry Marsh's group at UC Irvine also showed that there was a correlation between the level of cyclin B1 expression, which is a protein involved in cell cycle, and these perinuclear, so these surrounding the nucleus Huntington aggregates that are present. And from our previous work as part of the HD-iPSC consortium, looking at gene expression changes between HD and unaffected iPSC neurons, they found that there was changes in the nervous system development. And so this was kind of a little bit reassuring. And then later on, Sandrine Humbert's group came out and said that there was an issue, there are alterations in neurodevelopment in Huntington's disease. And this was a very interesting and groundbreaking paper that showed this, that there are these changes in neurodevelopment in Huntington's disease. But people are still able to live for a good amount of time with the Huntington's disease gene. And so this paper from Sarah Tabrizi shows that there is, Sarah Tabrizi's group shows that there are individuals are like cognitively and psychiatrically the same as unaffected individuals quite before onset of like motor symptoms. What they found, though, there are slight differences in these biomarkers, such as CSF neurofilament and plasma neurofilament light, as well as a change slightly in the putamen volume. But other things in the brain don't seem to change this far from onset of disease. And so this is reassuring for us, which means that there is a point we can go in and treat and prevent disease from happening, hopefully. So going back to our work, we wanted to see whether what we were seeing is kind of mimicking this in a dish. So are we seeing this kind of issue with neurodevelopment in our cells? So what we found, and we kind of extended our protocol to include additional cell lines, and we found that the level expression of this one protein cyclin D1, which is part of the cell cycle, was upregulated in a lot of our cells that were HD. And so we found that in our 50Q, so cell line containing 50 CAG repeats, there was an increase in this cyclin D1 protein. We also found that there was an increase in expression of a protein called OCT4 and nestin in green on the right in our HD samples, which kind of makes you think that they may be a little bit immature in their differentiation, in their like progression to becoming a neuron. And so now we saw that this was present in multiple cell lines. We quantified it by doing flow cytometry, where we can assess a lot of cells at once and assess their expression of proteins. We were able to see whether there were these, why was this happening, and whether we could prevent it in any way. So we had a look at single cell RNA-seq and used our an adult onset HD line and one unaffected line, and found that the cell type that was persistent in our HD samples was potentially a neural stem cell or neural progenitor cell. So we did the single cell RNA-seq, and the results we get are here on the screen. And each of these dots represents an individual cell, and how close together they are shows how similar they are in their gene expression pattern. And so if they're further away from each other, they have a different gene expression pattern. What we found was this green and brown population here were pretty much all HD cells. And so we wanted to figure out what they were, and they were these neural stem cell neuroprogenitor cells. So then Dr. Hernandez did a dig into the literature and found that if we potentially inhibit a pathway called the Wnt pathway, we would maybe be able to target this. And so we did the experiment where she treated with this drug called ICG, which inhibits part of the Wnt pathway, and found that when we looked then for this expression of this protein cyclin D1 on a Western blot, so looking at these red bands, we see that it's present in the 50q and the 53q when they're treated without drug, so with DMSO on the left. But when we then treat with this ICG drug, we're able to get rid of this cyclin D1 expression. So we're trying to prevent this from persisting in our cultures, and it worked. So kind of changing gears a little bit now, I want to talk about another project that was going on in the lab around about the same time, and talk a little bit about mitochondria. So mitochondria are the powerhouse of the cell. They are able to produce all of the energy required for the cell. And this is kind of their main function. And so the mitochondria have these folds within their internal membrane that form part of their function, provide more surface area for things such as producing energy. So what do we know about mitochondria in Huntington's disease? We know that mitochondria are affected in Huntington's disease. We have seen many papers over the years, these are just a few of them, that show that there are deficits in HD mitochondria and HD energy production, for example. Also the fact that this kind of might be tied to the fact that patients aren't able to maintain weight as well, because there is this bioenergetic deficit. And so we wanted to look at whether we could examine the mitochondria more. And we were collaborating with a group at Stanford University, or SLAC, and they use something called cryo-electron tomography to look at these small structures inside the cell. So cryo-electron tomography allows us to get a biological sample, a cell or a tissue. This is frozen in a very special way, and thinned to a really thin, to allow the electron microscope to image it. And this process of freezing is very special because it allows us to keep the native structure of the sample in good state. And this was done in collaboration, like I said, with people at SLAC, and Dr. Gong-He Wu is a fellow HDF postdoctoral fellow. And so we have this massive microscope here. This liquid nitrogen tank on the left is kind of for scale. It's around about the height of average person, like 5'5", something like that. And this is a very big machine. Ironically, what we actually plate the cells onto is a very, very small disk. And so it's these two extremes of how we can then measure differences in these cells. And so what we get with these microscopes is something like this. It's kind of looking through a sample. So say you have a sample and you can go up and down the sample, just looking through it as the way the microscope tilts the sample to get data. And so this is kind of going from bottom to top now. So we're going through the sample. And here we can see a mitochondria in the center, along with, as well as some tubules, so part of the cell skeleton, running through kind of like train tracks, going through the sample. And then you can kind of get like an averaged image at the end of it that you can see here, where we have the mitochondria in the center, and then we have those lines going through the parts of the skeleton. And so I spent a lot of time trying to optimize getting these samples for Gong-He, and we did this workflow to try and change how we could do it to make our samples better. And so we applied our normal protocol. We changed a few little things, and we were able to grow these cells on these grids. So this grid in the center here that's gold is actually very tiny. It's around about maybe 0.5 centimeter in diameter. And so that small, tiny thing goes into that massive microscope. It's very interesting. But we're able to see here on these cells, on these grids, that we can see the cells as they would grow normally. We can see the small parts of the cells, like the neurites, the extensions from the cell body itself. And this is how these cells would look just before harvest. So they look really mature. They look like neurons. So they're able to communicate with each other, and they're able to, they look like neurons. And so we're kind of happy with that. And then we would fix these samples in this special way by plunge freezing them into liquid propane to try and maintain that actual structure that we see in as close to what they would be when they're alive as possible. And so what we found with Dr. Gong He was that the HD iPSC neuronal mitochondria contain these enlarged granules. And these are electron-dense granules, and that's how the electron microscope is able to find them. They're able to see them more. And so in the top two panels here, we have unaffected individual samples. And so we see that the mitochondria outlined in yellow on the right, and then with the light blue to show the cristae membrane, so the inner folds of the mitochondria. And we see these dark blue granules in them, but they're kind of small. However, when we go to panels C through F, we can see that these granules become a lot larger. Or in the case of the Q109 samples, so this is a HD sample with 109 CAG repeats, that actually we don't even really see these granules so much, but the mitochondria themselves do not look healthy at all, because the cristae membranes or these inner folds of the cell in the mitochondria are not, do not look healthy. And so we were curious as to what these granules were, because it seemed that they were only apparent, or they were accumulating or aggregating more in HD. And so these mitochondrial granules, we found out were RNA granules. And how we found this out was looking at using a protocol called mass spectrometry. I was able to isolate mitochondria from our neurons. And then we sent these samples to Ranan Avenir, Dr. Ranan Avenir at Dr. Judith Friedman's lab at the time. And he was able to perform this mass spec experiment where you look at the differences in the protein content between the two samples. And so what he found was that there was an increase in the amount of proteins in HD, or an increase in representation of the proteins that in HD samples that were related to RNA binding. One of the RNA binding proteins that is involved in mitochondria is called GRSF1. And we found that if we reduce the amount of that GRSF1 in our HD mitochondria, we were able to get rid of or prevent these granules from forming. So kind of taking another little detour right now, mitochondria are actually very important for supporting the synapse. And we wanted to go into another project that I was working on that showed some promise for interacting with the synapse. And so I'm going to go quickly through this thing called SUMO modification. This is a modification of proteins that happens all the time in our cells. And it's a process which allows proteins to do their job properly. So there's proteins are modified all the time. And so we know that this can help change what happens. It's kind of like having a little badge on, like a little security badge, and or an access all areas badge, and being able to go backstage for a concert, for example. If you don't have that SUMO tag on you, you might not be allowed into the backstage area. And so that would be like being not allowed to get into the nucleus, for example, of the cell. And so this process is very reversible, and it keeps cycling. And so you'll have SUMO proteins added and removed, and it's a very transitional process. So how does this relate to Huntington's disease? Huntington protein is SUMOlated, and this was found by Joan Steffen, Dr. Joan Steffen, who's a very close collaborator of our lab at UC Irvine. And she found that SUMO1, one of the SUMO proteins, modifies Huntington, and this stabilizes it. And it also prevents these like aggregates from forming in HD. If you prevent mutant Huntington accumulation, it reduces neurodegeneration as well. And she found this in both fly and cell models. Then a graduate student in our lab, Jackie Geyer O'Rourke, she went on to study how Huntington was simulated, SUMO modified. And so she found that Huntington was SUMO modified by two different types of SUMO proteins, SUMO1 and SUMO2. And she also found that another protein called PIAS1 was responsible for modifying the Huntington protein. This project then got passed on to the next graduate student, Joseph Ochaba, who showed that if you performed experiments on mice, so we had HD mice, and he injected them into the striatum with a drug that would prevent the expression of PIAS1. And what he found was if we did this in HD mice, we were able to increase the amount of protein expressed that was related to synapses, this synaptophysin protein. He found that there was a reduction in the Huntington accumulation in HD mice, and it also restored RANGAP1 localization, which would hint that he's maybe restoring some of the nucleocytoplasmic transport deficits in HD, which we see there's a change in the transport of proteins between the nucleus and the cytoplasm. And so this is very promising data from the HD mice that he was able to show. And we wanted to take this now to a full length Huntington mouse model. And so this then got passed down to the next graduate student, Dr. Eva Morozko now, and she found that if you did the same experiment, so injecting into the striatum of these mice, what we were able to find was there was an increase, there was a rescue, in the synaptic gene transcription in HD. And so we're thinking that maybe there's some kind of support provided, or a rescue of the synaptic gene transcription, which meant that you might rescue some of the synaptic deficits that we see. And at the same time, I did this same kind of experiment. So I had our stem cells unaffected in Huntington's disease stem cells. And then I did an experiment where we put a PIAS1 siRNA, so it would prevent PIAS1 protein, making PIAS1 protein in these cells. And we found that this kind of had the similar effect of changing this gene expression to change the synaptic gene transcription that was very much similar to what Eva was seeing in the mice. And so with all this data pointing to the synapse and knowing that mitochondria are very important at the synapse, we wanted to kind of go back to our collaborators at SLAC and look at the cryo-ET, or have a look at the mitochondria. And at this point, I used a CRISPR modified PIAS1 line. So this was a heterozygous knockout. So we only had one then copy of the PIAS1 gene. So in theory, you should be only getting about 50% of the PIAS1 protein being made. And we looked at this in two HD samples in Q53 and Q66. So in these two samples here, we see that there are very much what we've become accustomed to seeing in these HD mitochondria, these enlarged granules. However, in the samples where PIAS1 was reduced because of this heterozygous knockout with CRISPR, we found that there was a prevention in these granules from forming. And so this points further to a potential therapeutic target being PIAS1 reduction. And so this is really great data. However, the brain does not exist in 2D, and this is all of the data I've been showing so far on the stem cells has been all in 2D. And when I say that, I mean it's been grown on a dish and plated on a dish. And so we want to look into the future of what stem cells can bring us. And one of the things I'm looking at right now is looking at these striatal cortical assembloids. What I mean by that is we have a striatal organoid forming and a cortical organoid forming. So forming those two, kind of mimicking those two parts of the brain. So the striatum that's affected in Huntington's disease, as well as the cortex that's also affected, and trying to bring those two together, and so that they can form a mini circuit of what is replicated in the what is represented in the brain. And kind of just to have a look at how these look in the dish, we can see that at an early stage, we have these like little balls of cells that are forming, but there's not really much organization to them at that point. And when we look at these actually in the dish, when they're a little bit later and a little bit more mature, we can actually see them with our, like the naked eye. We can see these organoids growing. And when you can see the assembloids as well, they kind of join together and make an even bigger sphere. And so when we look at some of the gene expression changes between HD and unaffected samples, there's a long list of these results basically from these changes in gene expression. And this is just a kind of a quick list to give you an idea that we're seeing a lot of these same things that are affected in HD, a lot of synaptic terms that basically would point to this being something that kind of like, we know this goes wrong in Huntington's disease in our samples. So the fact that we were able to replicate this in our organoids is very reassuring. And we're able to also measure this electrical activity that's present in these organoids. And so this is an example here, a little video showing how these organoids, when they're plated onto electrodes, how we can measure their ability to fire and communicate with each other within the cells. And so we can have a look at how many spikes, how many communications are happening between unaffected and HD organoids. And what we've actually found is our cortical organoids act a lot like what we call primary neurons. So these are mouse neurons that have been taken from the striatum or the cortex, for example. And so it's encouraging to see that these are actually behaving like what we see in those cells, because it means we're on the right track to making something that mimics the cortex. And so kind of looking still further into the future and how stem cells can keep helping us for HD, it's actually been very rewarding to do this talk, to kind of look how far we have come in my fairly short career of just over 10 years now. They've allowed us to further understand disease mechanisms, and I'm excited to see what we learn next. And so just to kind of summarize what I've shown you a little bit here, we know that stem cells have shown that there's issues for neurodevelopment in HD. There's changes in synaptic and how the neurons communicate with each other. That we've shown that there's issues with the mitochondria and how they're able to, how their structure is changed. This Huntington aggregates, we've seen other papers have shown that there are these changes. Dr. Hernandez, as well as Dr. Ryan Lim, had shown that there's changes in the brain microvascular endothelial cells, as well as all these changes that are present in the nucleus. And so we can go into the future now looking at whether we use organoids or kind of almost going back a little bit in time of having a mixed culture, but hopefully now we're able to do it in such a way that we can control the proportions of cells that go into that culture and be more definite and definitive with how we culture these cells, so that we're hopefully able to more resemble what happens in the brain and what happens in HD. And so with that, I would like to thank everyone in the Thompson lab, as well as all of our collaborators through these projects. Finally, thank you to the HD patients as well for all of their donations to make all of this research possible, because without the iPSCs, I wouldn't be able to do any of this research. So thank you very much.
Awesome. Thank you so much, Charlie. Thanks for sharing your work. That was a lot of work. As someone who used to grow stem cells, I know that is a lot of work. So we have a great response of questions, but please feel free to keep typing your questions into the Q&A box. As we start to go through the Q&A session, we will try to get to as many as we can. So I think I'm just going to start at the top and we'll go to the bottom. So the first person asks, are there places that focus on stem cell research in Huntington's on the US East Coast?
Yeah, I mean, yes. It's hard for me to think right now of specific people, but a lot of the people from that consortium were like on the East Coast or West Coast. I'm
I
sorry,
think I
my
might add
geography
to
is
this.
terrible.
Why are there so many iPSC labs or stem cell labs on the West Coast?
I mean, so there's one main reason. I mean, without getting into politics, but because of the ban on embryonic stem cell research back in the early 2000s, the California Institute of Regenerative Medicine was formed to be able to make stem cell research possible. And so that is one way that has funded a lot of labs across the West Coast. And so that's one. So that's why there's probably more stem cell research on the West Coast, but it is happening more and more on the East Coast now.
Awesome. So there are a fair amount of questions that are curious about the use of stem cells for therapeutics. And so I know your work and your talk focused on using stem cells mostly as a tool, but maybe you could also kind of give an update on if there's research out there that might be using stem cells as transplants or kind of where that research stands.
So there is the stem cells for HD working group consortium. I'm not sure how it's fully officially called, but stem cells for HD. I think they might have a website as well, and they are pushing for kind of what is the best cell type that we could use for transplants. What's the best time? What's the they're trying to cover the gambit of how can we make a really good therapy for HD? And it's based off a lot of the work that's, I think it was stem cells for PD, set up by Dr. Roger Barker. And it's kind of using that framework and then trying to make it more relevant for HD. And yeah, how can we do that? Because we know it can potentially be a great therapeutic based off of early fetal transplants and things like that. But obviously there's issues with that, with availability and ethics. And so, yeah, I think stem cells could easily be hopefully be a great therapeutic, and probably in combination with something like Huntington lowering or other lowering strategies that could come into play soon. Yeah.
Awesome. So that kind of gets at this question. Do you believe stem cell therapies could be used concurrently with other treatments such as gene therapy, gene editing, small molecules, and ASOs, both while it is in trials and after regulatory approval?
I mean, I think almost I mean, I'm not sure about in trials because we need to know that one's going to work first, I think. So we need to assess that individually. And then there can be trials that are done to bring things together. But I think almost it's going to have to be in combination because the stem cell therapy idea is only going to work at replacing circuits or helping preserve circuits that are already there. It's not going to change that there's still mutant Huntington protein in HD. And so I think we need to have this combination at least of maybe Huntington lowering at the same time.
Yeah, I agree. So there's two questions that one talks about where people would need to go to get stem cell therapies for HD, and another talks about how frequently patients would need to get treated. So I just want to be clear. Maybe you could talk about where these transplantation studies are. Are they in mice? What do those look like? And are they in people?
So nothing is being done yet in people. There's maybe a clinical trial on the horizon soon, and that will probably be maybe the first one that will come out, and that's going through the motions right now to get to a clinical trial. But everything so far has been done in mice, a little bit in higher animals, just to kind of get dosing and things like that correct. But yeah, nothing is in humans yet. And yeah, I do not advocate going anywhere that's not an FDA approved or clinical trial that is, for HD research, for HD basically, because nothing is proven yet for treatments.
Perfect. Thank you for clarifying that. So this next question is long. So it says, in recent trials and preclinical research currently, early intervention seems to be the goal for nearly across the board for HD treatment. Do you feel similarly about stem cell therapy? Do you believe it would simply be more beneficial to intervene early and then later stage patients would still benefit, though maybe less so? Or is there potential for intervention at later stage patients to receive negative effects similar to what is seen in trials for tominersen in 2020 and 2021?
So I think in my mind, I think stem cell transplants, for example, would be maybe later because like the data I just showed from Sarah Tabrizi's group, there's not much loss of the striatum like far from onset, which is maybe where we should be looking at closer to that time point for things like Huntington lowering. So I think it could be something that's a lot later, that would help in like maybe replacing lost circuits or just helping maintain the circuits that are still there in the brain. I mean, it could potentially work as a early therapeutic, but I think this one could be something that is a little bit later, and because we also don't have much room in the brain if we are not losing much space. If we're not losing much cells there, we need to have space to be able to put the transplant in as well. So
excellent, thank you. So I think let's take a little departure from the kind of clinical related questions and we'll do more molecular stuff. So an anonymous attendee asked, does the size of the CAG repeat change when HD-iPSCs are differentiated into MSNs? And then Sam Frank added on to that, do you see somatic expansion in any of your work?
I've not specifically looked at anything to do with somatic expansion. However, for example, our 109 line is not really 109 anymore. Just through culturing, as stem cells, it seems to expand. So it is kind of that is a kind of a good cell line to be able to use, and right now we're trying to CRISPR edit that line so that we can look at somatic expansion with the PIAS1 knockdown, because we've had some preliminary data that shows that it could potentially work for that, or at least help a little bit with the somatic expansion. So yeah, that could be something we're looking into. That is something we're going to be looking into with that line at least, and yeah, there could be CAG expansions that we see in culture.
So related to your 109 line, an anonymous attendee asked, the 109Q cells don't have the RNA granules in the mitochondria. Why do you think this is? Does the RNA-seq data suggest they don't have changed expression of the key genes you mentioned and which you knocked down later?
So interestingly enough, we haven't really seen much transcriptional dysregulation of the 109 line. I'm not sure why that is. We don't see the same gene changes. And so that could be part of it. It could be that this 109 line, I think the individual it came from was very, very young, like pre-10 years old. I can't quite remember exactly the age. But that was, someone who was very, basically they had a very bad CAG repeat, and it was detrimental, very detrimental. So I'm not sure whether we're missing something even with it being so progressive, and kind of like with the RNA granules, it's like were they there maybe when they were like neural progenitor cells, but we're looking at neurons because these mitochondria just do not look healthy at all. And so that could be what we're seeing here, is we've just missed that stage of the granules because we looked so late, and we're going to try and look earlier as well.
There's another question about the penultimate CAA repeat. So an anonymous attendee asked, there are some science that talks about the differences between CAG repeats and CAG-CAA and the severity of HD and the onset of disease. How does iPSCs mimic this? Do these iPSCs have CAG only or CAA and CAG?
We haven't actually sequenced our lines to determine that CAA, CAG, so I'm not sure. I mean, that obviously would be a very interesting thing to look at, so we can correlate that maybe with some of our phenotypes that we're seeing in the cells. But yeah, we don't know yet what the CAA, CAG is in our cells.
There's a question from Maria Cristina Capizzi. She says, really interesting work. Here's Maria Cristina from Humbert Lab. In the cryo-EM of the 109Q, mitochondria and the microtubules disappear from the imaging. Do you think this is an artifact, or could it be possible that the mitochondria lose their microtubule attachment?
Oh, interesting. I mean, that could be what it is. I think, yeah, those mitochondria are just so sick that we're not even, and we've even shown in the rest of that paper that there are these what we think are autophagosomes, so double membrane bound organelles that are containing a lot of the cellular waste. And we see what we think are mitochondria in those because you can kind of see the cristae and things like that. I'm not sure whether they're in those 109 line. We don't even really see much of that as well, because it could be that, yeah, we're losing this microtubule attachment, we're losing the mitochondria losing their structure, and losing their function as a result. And so it could be an interesting thing to look into. Thank you for that.
So Megan Donaldson asked a question about your PIAS1 stuff. So she says, one of your later slides showed a long list of gene expression changes that take place in HD cells versus healthy cells. Does the knockdown of PIAS1 in HD cells reduce these gene expression changes? You had mentioned that knockdown shows the reduction of granules in mitochondria, but what does that mean from a functional phenotype standpoint?
So we're still exploring the RNA granule and the mitochondria phenotype. So we're trying to dig into that more now to see what can be changed, and kind of getting a little bit away from the PIAS1 stuff, just to really nail down what is going on with these mitochondria. But going back to the PIAS1 stuff, it's kind of complicated with the organoid gene list, trying to figure out, if those changes are the same. It seems like some of the PIAS1 knockdown stuff is having an opposite effect to those gene changes in HD, and some of it's going the same direction. So it's still too complicated, and I didn't want to get into that too much for this talk, but yeah.
Awesome, Charlie. Thank you. We have a lot of questions that we're not going to be able to get to because we're at the end of our time. But I want to wrap things up because we are at 1 o'clock, and I want to thank you again. If you had questions that we didn't get to, I will send those to Charlie, who graciously said she is willing to follow up with all of you. So I want to end by again thanking our sponsor, uniQure, as well as everyone who has donated to the HDF to help support the research that we fund, our audience, and last but certainly not least, Charlie, thank you so much for joining us today. This is the last webinar in our 2023 series, but I'm working on an exciting lineup for 2024. We are going to kick things off in January with a webinar where I will recap HD research past to present. So covering from the discovery of the gene through to ongoing clinical trials. So stay tuned for more details. Thank you all so much for joining us today, and we hope to see you at our next webinar in 2024. Thanks, Charlie.
Thank you.